Hi, second video that needs to be corrected. Okay, mutagenesis. What causes mutation? It can be caused by physical changes. Physical means if you are exposed to X-rays. You go to X-rays, right? In X-rays you have all these particles penetrating your cells, and they actually cause mutations by if they hit the DNA, it will actually cause a break. But because the dose that is being used is very, very low, so it's considered as safe. But you don't want to be staying in that room for unnecessary reasons. I mean, that's why if you go into X-rays, you'll find that the operator will go behind the screen because they don't want to get exposed. So those are physical, and you can also get mutations. These are chemicals that can change the qualities of DNA. It can break the DNA, it can cut the DNA, it can change the property, the binding properties. We know that A will bind with a T, C will bind with a G all the time. But there are chemicals that can change the properties of a C, that now it suddenly is more prone to bind to a T instead of a G. So those are and then of course enzymatic methods. Some enzymes in the body itself are prone, sometimes they can make changes. Now mutations can either be spontaneous or induced. How do I describe that? Spontaneous means, well, of course it's beyond your control. Your cells undergo replication, right? They make copies. So your polymerase sometimes makes mistakes. So these are spontaneous mutations. You don't control them. That's why we need an efficient repair system. But induced mutation are when you are exposed to the environment. For example, if you happen to live beside a nuclear reactor, and then there is a leak in the nuclear reactor, and then you what happens in Chernobyl. So you get exposed. So these are induced mutations. So that means it's trying to say that when the cell makes or errors in the cell that causes it to become a mutant, that one is called spontaneous. But something that you add or you expose your DNA to and becomes, so that is induced mutations. Now things that cause mutations are called mutagens. All right. And here are the types of mutations. Remember, mutations can result in a substitution, a change, or deletion, or insertion. Now, deletion and insertion, we saw that it will cause frame shift. Remember, it changes the reading frame. But substitution, it depends what happens. Now, here's what so this is the difference in substitutions. Now, when you have a substitution, it can either have the same meaning. Why does it have the same meaning? Because some codons have more, some amino acids have more than one codon. I give you an example of a proline. We know that CCC and CCU both codes for proline. So if there is a mutation between CCC changes to CCU, it still codes for proline. So this is what you call as silent mutations. Let's go back to the look at this. So if CAU, then this U mutates and changes into a C. So you have a CAU changes to a C, so it's a point mutation, a substitution, but it doesn't change the meaning. Right? So this is what we call as synonymous mutation. Okay, let's look at this methionine, because there's only one codon for methionine. So if it's AUG, if you change it to AUA, does the meaning change? Yes. If the meaning changes, then it is not a silent mutation. It means something else. So it's either so that is a missense, right? So we're going to look at that. So if there is a change and the change results in the same meaning, so it's silent, or it's also called synonymous. If it's giving a wrong meaning, I'll give you an example just now. methionine AUG becomes what? AUC, isoleucine. That one is a missense. There's also mutations that results in no meaning, and that's called a nonsense. Here, this is the original sequence, CGA. CGA codes for arginine. If the first C, right, if that C mutates into a U, then what you get is a UGA. And what is a UGA? UGA is a stop codon. So that's where you call it a nonsense. These are just examples, yeah? Right? It's not that this particular sequence will change that. You know, it's just example. If. So if it changes into a U, then you get a nonsense mutation because UGA is a stop codon, and so you call it as nonsense mutation. If the G changes into an A, is it transcription? Is it a transition or transversion? G changes to A. It's a transition because this is a purine and changes into purine, so this is a TS. TS is transition, TV is transversion. So if the G changes to A, this is a missense because CGA codes for arginine, but CAA codes for glutamine. And then if, for example, the A changes into a U, now this is an example of a silent mutation because CGA codes for arginine, CGU also codes for arginine. Okay, so I hope you can see the difference in what this all mean. Now, this is what I said, it's easier to see what this means. So let's say you have the original sequence, the bed, red, dog, and the cat. So this is original sentence. If this is an example of what is this? Is it a frame shift? Is it a missense or nonsense? This is actually a missense substitution because bad has been changed to sad. Similarly, dog has been changed to doc. And these are two missense mutations here. What about this? You remove the D from the original sequence, and then you get from the bad, red dog, and the cat becomes the ba and organic. So that is a deletion. Or if you add an N, you put an N in, it becomes the bandrid dog, right? Sound like somebody from the Game of Thrones. And so this is also a frame shift. So it changes the meaning altogether. So that is called a frame shift. So here, missense, deletion that's causing a frame shift, and addition that's causing a... In the real example here, it says, so you've got the wild type. Wild type is something that is normal. Wild type is called wild. It's a general term in genetics. So you have ATG codes for methionine, TGG tryptophan, GCA alanine, GTG valine, CCT proline. So that if the first A changes to a T, it still codes for alanine, so that's why it's called a silent mutation or synonymous mutation. If that G becomes an A, it becomes a nonsense, it's stop codon, so now it becomes a nonsense mutation. It totally, that means the original protein, if it's that long, because there's a mutation, there's a stop codon, then what you get is a small protein, alright, it's called a truncated protein. Then you have, if it's that A, if that G changes to C, it becomes from tryptophan to cysteine, then this is a missense. If you add a G, see how the meaning changed from methionine, tryptophan, alanine, valine, proline, it becomes methionine, tryptophan, glycine, serine, and alanine. And similarly, if you minus 1, then methionine, tryptophan, arginine, cysteine, leucine instead of the original methionine, tryptophan, alanine, valine, proline. So I think that wraps up the mutation lectures. Alright? So I'll see you in the next video. Bye-bye.